So before I start, this is going to be the fourth time that you're going to hear a similar story here. And pedagogically, I think it's important for you to check if the stories that Wendy, Marcus, Daniel, and myself were telling you, whether they're consistent among themselves or not. So which part of the story you relate the most to. So here's my take on the spin flavor of quantum biology. This and okay, wait, how do I go back? Back. Awesome. Okay. I like to call myself a quantum engineer. So this means that I build instruments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything around us. So people do get a little bit confused when I say that I study things that might be going on inside birds, inside butterflies. So before I explain where I'm going, I really need to tell you where I come from. So we're going to start by talking a little bit about hardcore quantum mechanics. And by the end of the talk, we will have talked about things of biological relevance, such as organismal migration, how our cells respond to oxidative stress, to radiation. And I hope I will have convinced you that I think we can learn with nature on how to build better technologies. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. I usually start my talks by saying that I think humankind is obsessed with measuring things better because this might mean that we understand them better. You can, for example, measure better frequencies. This is an atomic clock that sits at NIST, the National Institute of Standards and Technologies, and that defines very precisely what a second or what a hertz is. Based on atomic transitions. We might think about measuring better magnetic fields so that the image of your baby is better resolved. And yes, that's the magnetic resonance image of a baby inside the mother's belly. Or we can think about measuring better accelerations with those tiny little accelerometers so that your gaming experience is enhanced. And those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. And actually, you can mathematically prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. In my past, I worked with a technological quantum enhanced sensor, and I'm going to spend a couple of minutes talking to you about it for a very particular reason. I really do not care about this technological quantum sensor anymore, but I'm going to argue later on in my talk that it works exactly as some quantum sensors made by nature. So here's my talk, and here's the promise. The promise is that we can put a single spin in diamond, in the material diamond, to work as a very sensitive quantum enhanced magnetic field sensor. So spin. For those, I travel with my props. I never leave home without my spins. So spin is a fundamental property of matter. It's as fundamental as mass and charge. And in the same way that charge measures how much an object interacts with a gravitational field, charge measures how much an object interacts with an electric field, spins measure how well an object interacts with magnetic fields. And in the same way that charge can be positive or negative, spins are usually represented with an arrow, and this arrow can be up or down. And again, it's all an abstraction. Arrow up or down mean different energies of interaction, say, with a magnetic field. This particular spin that I worked with in the material diamond, it is actually a trapped electron that has a spin. So in the material diamond, there is a particular lattice defect that is equivalent to a trapped electron. Okay. And this trapped electron is extremely interesting because it is what's called a color center. This means that if you shine light at it, it will absorb this light, get excited, and then emit light back. Okay. For reasons that that are only going to make sense why I'm mentioning this later on. This defect in diamond has a very nice property, which it is that it has intensity dependent, fluorescence intensity dependent quantum state. This means that if I shine light onto such a defect in the material, it's going to absorb light, get excited, and emit light back, but it will emit a different amount of light if the spin is up or if the spin is down. This means that if you look at the screen of your computer and see how much light is being emitted, you can make a quantum measurement of the state of the spin, up or down. But I promised that we could put it to use as a very sensitive magnetometer, and here's how that works. To the right there, I put two different energy levels. So in this y-axis here, it's different energies, and state marked 0 and 1 are just, say, spin up and spin down. And their differences in energy is about the microwave range, about 3 gigahertz. And it turns out that the state that I marked as 0 is insensitive to the presence of magnetic fields. This means that if I immerse my diamond material into a magnetic field, state 0 will not care. However, state +1 is sensitive to magnetic fields. This means that if I immerse my diamond in a magnetic field, state 1 will be promoted by a tiny quantity called delta. And this delta, by something that's called the Zeeman interaction, is proportional to the magnetic field strength. And if you're sleeping, that's a good time to wake up, because what I'm going to tell now is actually extremely important. It's a point that doesn't usually get made. Okay, here it goes. The problem of measuring a magnetic field is actually mapped onto the problem of the experimentalist being able to measure a mismatched delta from a known energy difference in the absence of magnetic field. And this actually underlies 90% of all the modalities of so-called quantum sensing. You have a certain energy difference that you know. This energy difference gets tweaked a little bit in the presence of a quantity of interest, say a magnetic field, an electric field, or a temperature. And being able to measure this tiny difference in energy actually gives you indirect information about the quantity that you care about. This underlies all quantum sensing. In fact, it's a signal processing problem. Engineers and physicists know how to measure mismatches, detunings from resonances for many, many decades now. And that's actually super important, I believe. But for reasons that I will not have time to explain, this modality, like using those properties as, using those energy levels as a way of measuring tiny quantities, only works while the spin is well described by the laws of quantum mechanics. Everything that starts quantum dies classical. After a certain time, the electron spin will no longer be well described by the laws of quantum mechanics. It will be better described by the laws of classical mechanics as a tiny magnet. And this way of sensing tiny quantities is only valid, is only quantum enhanced before those before the quantumness has decayed. And for Daniel Felinto here, or for the experts, I'm going to make a parenthesis. The idea here is that you put the states into a superposition, and basically you run a Ramsey experiment on it, and a difference in phases picked up translates to a difference in population that you can then read. So this only works because of superposition. Okay, I'll close the parenthesis for the experts. So, but the big picture here is that you have a tiny little sensor that can measure tiny little quantities. And I have just shown you one example of a technological quantum sensor that works at room temperature and under messy conditions, the messy condition of the diamond lattice or the sample that it's in contact with. Okay, and that's going to be very important for what I'm continuing to talk about. So up to 5 years, like until 5 years ago, I was used to dealing with this type of sensors there, like all humankind made sensors. But at some point, almost by chance, I realized that nature made sensors that overperformed humankind made sensor in crazy, crazy ways. Here, I would like to talk to you about a nature-made magnetic sensor that I'm going to argue is very similar to the technological quantum sensor of magnetic field that I just shown to you. And in order to do so, we're going to have to start with the biology at the nanoscale, namely the chemistry. So, in test tube chemistry, it's been known for many decades now that there is a class of chemical reactions that depend on spin. Again, there is no doubt about that. This has been known for many, many decades. In a nutshell, here's how this works. There's a chemical reaction happening, and at some point, this chemical reaction comes to a crossroads. At this point, the chemical reaction effectively looks for the particular spin state of a particular electron. If that state, the spin state, is up, the chemical reaction continues through one path. If that particular spin state is down, the chemical reaction continues through another path. Importantly, the final products of those two branches are macroscopically different. So a tiny quantum property is known to affect big time the result of a class of chemical reactions. Now, if at the moment where this chemical reaction is at that crossroads, an external magnetic field interacts with this particular spin, it actually, via a process that is a bona fide quantum sensing process, exactly with what was going on in the spinning diamond, this spin is going to interact with the external magnetic field, and the external magnetic field, by being sensed, is actually going to alter the probability of the spin being up or down, so that the magnetic field is also going to be able to influence if this path is taken or this path is taken. Okay, so I'm simplifying the picture here because for most of the chemical reactions that I care about, that Wendy, Daniel, and Marcus we care about, it has to do with the quantum state of two spins. The two spins can be in a singlet state, meaning one pointing up, one pointing down, that is related to one chemical pathway, or they can be in a triplet state with both spins pointing at the same direction. But it's exactly the same idea. Spins and their interactions with magnetic fields are altering big time the final products of chemical reactions. Now, this has been demonstrated at room temperature, in the gas phase, in the solid phase, in the liquid phase, and for magnetic fields as small as the magnetic field of the Earth, which is orders of magnitude smaller than the magnetic field that you sense when you put your cell phone close to your face. Now, so how did the conversation migrate, pun intended, from chemistry to biology? It's all about birds. Okay, I really do not care about birds. I will go away from bird in a while, but birds have the big advantage that they were actually, they have to be given the credit that they brought the conversation from chemistry to biology. So birds are known to migrate, and when they migrate north and south, they do so unambiguously using the magnetic field of the Earth, at least as a partial cue. Again, tiny magnetic field. People didn't really know how birds were doing this stuff. And in 1978, there was this theoretical biophysicist who made this outrageous hypothesis at the time that went in this way. He said, well, were the same type of spin-dependent chemical reaction at play under physiological temperatures inside the birds somewhere, birds and organisms in general could sense magnetic fields to the extent that they could sense different physiological concentrations of products coming from those spin-dependent chemical reactions. Again, outrageous hypothesis, right? And then things like the following was proposed. And again, this is, I forgot to put the reference, but this is taken from Klaus Schulten's former website. And we've seen this picture before in Marcus' talk. So the idea is that this has not been either proven or refuted, but maybe that's how this would go, right? So imagine that you're a bird, and the bird moving their head left or right, seeing different magnetic field lines. Imagine if it were possible that a certain magnetic field to the left or a certain magnetic field to the right changed the product of a certain chemical reaction in your eye, for example, that could maybe photomodulate the sensitivity of your retina to photons. If it were so, the bird looking this way or this way would see brighter or darker, depending on the direction. Maybe this could explain, maybe birds would be able to see magnetic field lines. Again, just a hypothesis. The problem is that we don't really know how to do this with birds yet, which is part of the problem with this whole shebang, but we're going to get there. But experimental biophysicists got interested in this crazy, outrageous idea by Klaus Schulten in 1978, and they started looking for proteins that could sustain spin-dependent chemical reactions. And at that point, the only animal because birds, protein known to sustain spin-dependent chemical reactions was this flavoprotein called cryptochrome. Now, cryptochrome, again, is a flavoprotein. It has a flavin, so it's a fluorescent protein. It exists in the retina of birds, in the antennas of migrating butterflies, but it's also present in all our cells in our body because it also has circadian rhythm regulation functions. Okay, so cryptochrome can sustain spin-dependent chemical reaction. And it's very interesting because cryptochrome is extremely widespread throughout the tree of life. All those organisms there express cryptochrome, from diatoms to humans, to birds, to insects. And there is, again, correlative evidence for all those organisms boxed in red that magnetosensing might have to do with cryptochrome. Okay, and it turns out that after 40 years of research on that, I mean, not my 40 years, but 40 years of research in the field, there's really good evidence that cryptochrome-based magnetosensing is really a thing, but unfortunately at very disconnected length scales. To the left of my slide, you see what I call the chemical length scale. Okay, and I'm going to explain a little bit of that evidence in a bit. But for example, for cryptochrome or similar compounds in solution, there is absolutely no doubt that they are behaving as bona fide quantum sensors of magnetic fields in exactly the same way that the spin in diamond is. I'm going to explain that top left plot that appears in Daniel Katnig's beautiful Nature Chemistry paper. And here's what Daniel and his colleagues did. They put cryptochrome compounds in a vial. Okay, then they excited cryptochrome with the laser. Again, cryptochrome has a flavin. It gets excited and then de-excites, emitting fluorescence. And what they did was they tracked the fluorescence of those compounds as a function of the time during which you applied this laser. The first thing that you see is that fluorescence is going down. You're bleaching the chromophore, you're killing the chromophore. But as the researchers post a tiny magnetic field on and off, as you can see in the inset, the fluorescence is also modulated in the way that I'm not showing it here, but the fluorescence actually follows the field being turned on and off. Again, this is not an artifact. This has been demonstrated using different field frequencies, different field intensities, but this is in one-to-one correspondence to what I told you in the very beginning about the spin in diamond. For the spin in diamond, just by looking at how much light it was emitting, we could say if the spin was up or down. Here, it's actually the same. Just by looking at how much light is being emitted by cryptochrome, you can know if the chemical reaction continued through the triplet branch, or the singlet branch. Okay, this is, if you will, a quantum state readout for the quantum physicists. Except that we go from that very nice quantum chemical evidence to things that are absolutely macroscopic. We have all talked about the evidence that birds might be doing their things following in a way consistent with a spin model. Flies, for example, they're not known to migrate, but they can be trained to find food. They can be conditioned to find food based on the presence of magnetic fields. I have no idea how they do this. That's harder than quantum mechanics. And then the researchers knock out the cryptochrome gene from the flies, and those flies are no longer able to be conditioned to find food based on the presence of a magnetic field. In a further experiment, the researchers put back human cryptochrome in the flies, and the flies are back to being able to find food based on the presence of a magnetic field. But it's really hard, even though that macroscopic evidence is mostly consistent with the physiological response being driven by a spin mechanism acting under the hood. It's very hard to say that, well, the bird is doing its thing because there's a quantum stuff happening inside it somewhere, right? So here's the deal. Here's what we are trying to do in my group. We are trying to bridge those two length scales. Okay, we want to take a bottom-up approach where we try and take single cells and apply the same type of chemical or quantum protocols in order to study and, importantly, control those spins inside proteins as if they were, because they are bona fide quantum sensors. So what we're building are actually glorified microscopes with coils. And when I say that I'm building a microscope with coil, it doesn't look like the thing to the left. It looks like the thing to the right. None of those are my pictures. Those are internet pictures. But that is a setup from a quantum experiment. Okay, it's like a big optical table with a lot of lasers, electronics around. And what we are doing is building something like that works on a biological sample. For the physicists in the audience, I would call it a light matter interaction experiment where matter is biomatter. And I really think that this is the bottleneck of the spin flavor of quantum biology. Most experiments today have been done by chemists and biologists. Those experiments are necessary, but I'm arguing they're not sufficient because one cannot, in the same experiment, track the spin states and physiological consequences of different spin state preparations. And that's what we are trying to do with our specialized microscopes. So I'll skip a lot of things here, and I'm just going to say that this is not about birds. My talk would be so much poorer if it were only about birds. There's now consistent evidence that many other chemical reactions of biological relevance seem to be they are consistent with being driven by spin phenomena, from the regulation of the production of reactive oxygen species to yield of DNA repair to cellular autofluorescence that is magnetic field sensitive in a way consistent with spins. But usually the thing that makes people come to my camp is this last picture here, which is not mine, which is from my good friend Peter Vierlinger from Munich. So Peter Vierlinger is a bona fide precision measurement physicist. What he does for a living is build very good Faraday cages, very good hypomagnetic chambers. Okay, so he started, and then he builds those very good Faraday cages, and then he shoves ultra cold atoms experiments inside to measure very precise things. But he started hearing things about magnetic field effects in biology. So here's what he did. He grew tadpoles under one of his hypomagnetic chambers for 2 days. So magnetic field of the Earth is about 50 microtesla. Inside his chamber, they have a noise level of about 1 nanotesla, so super tiny. He grew tadpoles for 2 days because that's as long as he could go without asking for a bio permit. Again, he's a physicist. And now this experiment has been reproduced by other groups, and they all agree with his findings. But here's what he did. Tadpoles, 2 days. If he put inside the hypomagnetic chamber a tiny magnetic field mimicking the magnetic field of the Earth, the tadpoles were okay. If he grew a new batch of tadpoles under only the 1 nanotesla magnetic field, the tadpoles were, as you can see, about 30% malformed. Let me recap what I just told you. We're not even putting a magnetic field. We're taking out a tiny magnetic field, and we're messing up with embryogenesis after 2 days. This opens up all sorts of Pandora box type of questions from, for example, if we want to colonize Mars, what's the magnetic field on Mars? Can we grow lettuce on Mars? Can we reproduce in Mars? Right? And again, because the magnitude of the fields involved here are so small, everyone's best is that this is actually spin driven. And if I would need to guess, I would say that the thing that gives the tadpoles their color is melanin, the same pigment that we have on our skin, and there's very rich spin physics in melanin. But this is the type of stuff that I think about. Another very recent evidence that is consistent with spins is the following. There's a couple of papers, one from John's group in Surrey, that have shown that by applying weak magnetic fields onto cells, you can actually mess up with ion channel functioning. Okay, so the idea is that if this is indeed true, that you can mess up with ion channel functioning with the application of magnetic fields, well, here's a complete new way of doing optogenetics. Nowadays, to do optogenetics, you genetically encode proteins that open and close ion channels if you turn a laser onto those. Here, what I'm proposing is the quantum version of that. Everything is already there. You do not need to genetically encode anything. And just by tweaking magnetic field parameters, magnetic fields penetrate very deeply. You could, in principle, open and close ion channels, maybe at will, once we find out how to do those things deterministically. And I'm dead serious when I say that in 30 years, where I want to be is to have my phone and say, well, today I need help with wound healing. You go to your app and then you press a button, well, wound healing. And then your field produces the exact magnetic field frequency and intensity that you need in order to do wound healing. And then you do this, except that in order to get, this is absolute science fiction today, but in order to get there, which is already accessible with crappy technology, you need this type of quantum experiments that we're trying to do for the very first time in the world. We need to be able to use those big optical tables to find the quantum map, if you will, of which type of magnetic field intensity and frequency mess up deterministically with each chemical reaction that depends on spin. So here's what we are setting up to do. And one thing that I didn't have time to explain, but I will mention anyway because it's impossible, is that for reasons that are well understood, the spin-dependent chemical reactions that are generally known, they depend on weak magnetic fields on the order of the Earth's, on the order of that produced by your cell phone. This means that if you put the magnetosensitive proteins that are not just cryptochrome, there's all sorts of other proteins that are now known to be able to sustain spin-dependent chemical reactions. If you put a vial of those proteins inside an MRI machine, a nuclear magnetic resonance machine, that 5 Tesla magnetic field is not going to alter the chemical reaction. Things that alter the chemical reaction are the magnetic field of the Earth, this guy here, right? So actually, we also need to understand exactly what those things are doing to us in the long run. I don't want to alarm anyone, but that's, we should start thinking about this. So this is what we want to do. We want to either establish or refute into a quantifiable way whether quantum physics is responsible for important physiological processes in biology, and importantly, can we start to learn how to deterministically manipulate it for, say, theranostics, right? And we're not the only people doing this, but we're definitely one of the only people who are actually trying to put quantum technology onto the game, bring quantum-inspired instrumentation towards harnessing the endogenous quantum mechanical degrees of freedom in biology. And I would just use 10 more seconds to say that I think we need this instrumentation. This is something different, but we also need to make an effort to bring quantum biology as a field to the mainstream by design. Right now, there is zero incentive for having young people enter the field, and that's the death of all new fields. So the other hat that I wear is the hat that tries to actually bring people to quantum biology, so that maybe by design in 10 years, there is a standing call for quantum biology at FAPESP, at the National Science Foundation in the US, right? And I think about things of the following kind. Well, if we want for there to be a quantum bio department in universities in 25 years, what do we need to start doing now? If you think about like bioinformatics departments, they didn't exist 25 years ago, right? So what do we need to start doing now if we want to be able to have a major in quantum biology in 30 years? So I would like to thank my people who are my rock, my lifeline, and we are battling together to survive in a system that we're not fit to inhabit. I'm very, very thankful to all those great scientists. And I'll end just by saying, may the quantum be with you.